the director of the CF clinic here, and so I have an interest in Pseudomonas infections, which is the predominant infection in the CF lung. And really the focus of treatment of cystic fibrosis is treatment of Pseudomonas, because it's an infection that we really can't eradicate. So it's an intense interest to figure out why that is. So the question then is, why can't we get rid of this infection from the CF airway? And the reason is that when we think of infections, we usually think of the bacteria as a single organism or a single cell. But that's really not how the bacteria sees itself. The bacteria sees itself as part of a community. And the community is called a biofilm. So what a biofilm is when a group of swimming bacteria decide to quit swimming and they attach to a surface. And subsequently, they actually lose the ability to swim. They lose their flagella and their pili, and they become irreversibly attached and develop a very compact, dense cell environment. And at that point, that's more or less what a biofilm is. Under certain circumstances, the biofilm can grow bigger and elaborate and form these large mushrooms that can actually harbor swimming bacteria in the core of it and can allow bacteria to escape and colonize elsewhere. Nothing like this actually occurs in the CF airway, but it's a fascinating community of bacteria. So biofilm infections are basically the infections that we can't treat with antibiotics, and it turns out that approximately half of the infections in the human body are biofilms, including infections such as dental cavities, chronic ear infections are biofilms, infections of the urinary tract, especially those that involve kidney stones or the prostate, and then infections of the heart valve or the blood vessels, infections that involve catheters and instruments, artificial joints, things of that nature. These are all infections that usually have to be surgically removed or drilled out, or are very, very difficult to treat with antibiotics. So Pseudomonas, which again is our prime focus in the CF world, occurs as a biofilm in the CF lung. There's approximately 30,000 people with CF in the United States, and 90% of them are infected with Pseudomonas. The median survival now is 36 years, which is a considerable advancement over what it had been even 10 years ago. The biofilms in the CF airway are located in these mucus plugs in these enormously dilated airways. The second Pseudomonas infection that causes biofilms in humans is contact lens wearers who develop bacterial keratitis. There's approximately 30 million contact lens wearers in the United States and about 30,000 infections a year. 70% of those infections are due to Pseudomonas, and it's actually the formation of the biofilm on the contact lens which is very important in the progression of the disease. And finally, severe burns are commonly infected subsequently with Pseudomonas. There's approximately 45,000 hospitalizations each year for burns, and about half of the ones that become infected with Pseudomonas, and in that group, the mortality is over 75%, so that's an extremely severe condition. So the reason the bacteria wants to form a biofilm is because it's an ingenious way of avoiding being eaten by the neutrophils and other phagocytes that are capable of ingesting the bacteria. So if they form a biofilm together, they're too large for the neutrophils to ingest. But there's actually more to it than that. It turns out that the biofilm state allows the bacteria to become resistant to common antibiotics such as tobramycin. And there's a number of mechanisms, I won't go into that, but it's a resistance that if you disperse the biofilm, the bacteria again become sensitive. So the biofilm is a very important way that the bacteria is developed to avoid being killed, either by the patient or by the doctor. So an idea that we have developed over the years is that part of the way that the biofilm forms in the human body, especially the Pseudomonas biofilm, is that it's able to take the actual cytoskeleton and the DNA from neutrophils that were sent to kill it. When the neutrophils die, it incorporates those polymers, and that's a way that the biofilm gets a head start with its development. And this demonstration of this is on a confocal chamber slide, in which if you look at Pseudomonas after 48 hours, you can see that they're forming sort of a biofilm on their own. But if you combine them with neutrophils in the same time frame, you get a very thick and dense biofilm. And if you look at a three-dimensional view, you can see how much taller and thicker and much more structure there is if there's neutrophils and neutrophil products present. So it's a way that the biofilm can use the host defense to its advantage. So we have an assay to rapidly screen for the ability of neutrophils to enhance the biofilm growth, and it's a 96 well plate that has these pegs that project into the 96 wells on the other side of the plate. And so we combine neutrophils and the Pseudomonas, and they form a biofilm on these pegs, and then we're able to stain with a dye that is able then to be read colorimetrically and quantify the amount of biofilm on each peg. And what we found then is, like I showed you in that slide, there's an enormous enhancement of the biofilm development in the presence of neutrophils. And what's really interesting is if you lower the concentration of Pseudomonas, so there's only 1 to 1,000 Pseudomonas per neutrophil, that's when you get the greatest enhancement. So it's a mechanism that actually enables a low number of bacteria to succeed in the presence of neutrophils. And this is a fold increase. And you can see at the lowest concentration of bacteria, there's a 400-fold increase in biofilm development. And then what's really interesting is that we can duplicate this without neutrophils just by taking the DNA from the neutrophil and the F-actin, which is the cytoskeleton or the sort of the structure of the neutrophil. We can combine those and form polymers of F-actin and DNA, and the Pseudomonas is stained here in the green. I don't know, maybe I can turn down that lights up a little bit. The Pseudomonas is stained in the green, and you can see that when they have this polymer of actin and DNA to latch onto, that's where they organize. And we can see the same thing when we lyse neutrophils. We get polymers of F-actin and DNA, and that's where the neutrophils, or that's where the Pseudomonas binds to. So that F-actin in the DNA is a scaffolding for the early formation of the biofilm by Pseudomonas. So what our grant was focused on was a method to disrupt this biofilm that's been enhanced by the neutrophil by targeting the F-actin in the DNA. And there's a drug already available called DNase, which is an enzyme that cleaves the long strands of DNA. It's commonly used in the treatment of cystic fibrosis. And then what we've done is taken long negatively charged strands of aspartic acid to combine with to the DNA and to unravel these polymers and put the Pseudomonas at a disadvantage. And so what we found is that early on, if you have a biofilm that's been growing for 24 hours in the presence of neutrophils, you see that the DNase causes quite a bit of disruption of the biofilm. And aspartic acid does as well. And together, there's not much of a combination. But if you let the biofilm get even thicker and harder to treat, you see the DNase has an effect. Aspartic acid doesn't have an effect, but there's a synergistic effect of the two. So if you use the two agents together, we're achieving a much greater disruption of the biofilm. And this might be the point at which an antibiotic could treat this infection. And if you go even farther and make a thicker biofilm and then reduce the exposure of these agents to a more relevant concentration, you see again that under concentrations in which DNase and aspartic acid by themselves have no significant effect, the combination has a significant effect. And we've been able to subsequently reproduce these using strains of Pseudomonas that are recovered from the CF airway, which are significantly modified from the laboratory strains of Pseudomonas. Just by frame of reference, if you take antibiotics such as ciprofloxacin and tobramycin, they have no ability to disrupt the biofilm. So taking these findings, we really aren't in the position to study them directly in the CF lung. So what we wanted to do was look at contact lenses as a first target for this technology. So again, there's a lot of neutrophils that accumulate at the site of contact lens infections, and there's about 90 million contact lens wearers in the world, including 30 million in the United States. And clearly there is biofilms. These are electron microscopy of the lens and contact lens wearers, and you can see how the biofilm forms on the surface. So using our model, we tested to see if combining neutrophils with Pseudomonas does enhance the biofilm growth on the lens, and clearly it does. This is a log scale, so this is comparing approximately 1,000 to 100,000 or 1 million in the presence of neutrophils. So neutrophils drive the same biofilm growth on the contact lens as it does in our other system. And we see the same result as well with the combination of DNase and aspartic acid. We get this synergistic reduction in the biofilm associated with the contact lens. So just in conclusion, the way that this technology is being developed, we've been using our 96-well plate screening method. We've been testing different combinations of agents to find out which is the best strategy to dissolve these biofilms in the laboratory. And now we've taken some of our best candidates and we're looking at dissolving the biofilms in contact lenses. And then starting this summer, we have a collaborator in Texas that's actually going to test these candidates in a model of rabbit contact lens eye infections to see if what we found using just the contact lens translates into a benefit in an actual animal model. And then finally, my lab is looking at the same candidate molecules in a model of Pseudomonas mouse burn infections. So this will be two relevant disease states in which Pseudomonas biofilms are the predominant infection that we can test to see if this strategy is a way of disrupting the biofilms and making it more susceptible to antibiotic treatment. And finally, if these two work, then we'll have the ammunition that we need to go back to the CF lung and try it there. So in conclusion, the biofilms that are persistent, we think occur always in the setting of abundant neutrophil accumulation, and that the neutrophil probably is an essential component of these biofilm formation, and that by disrupting the neutrophil DNA and actin, it's a new way of targeting these types of infections. And these are a list of all the collaborators here at the University of Colorado Children's Hospital that I work with. And thanks to the state for the support of this research.